welcome. Good afternoon, good evening for those who are online. Welcome to the 2025 Hepatitis B Foundation Baruch Blumberg Prize Seminar. My name is Shari Cohen. I'm the president of the Hepatitis B Foundation, and I'm very, very pleased to welcome everyone here. The Blumberg Prize is the Hep B Foundation's highest honor. We've been actually giving out this award, which used to be called the Distinguished Scientist Award, since 1997, and we changed the name to the Blumberg Prize in honor and memory of Dr. Baruch Blumberg in 2014. Each year, the prize is given to someone who has made an outstanding contribution to hepatitis B and the diseases that it causes. And we are thrilled to have our Blumberg Prize seminar today and our awardee. Just a quick update about the prize. So every year, the nominees are brought to the table by the president of the Hepatitis B Foundation, the president of the Blumberg Institute, faculty at the Blumberg Institute, and previous prize winners. And then all of the previous prize winners get to vote. And so it's very illustrious because this really is an award of peers. So in order to announce and bring up today's prize seminar speaker, I am going to introduce Dr. Jutao Guo, who is the acting president of the Blumberg Institute.
Yeah, it's a great pleasure to have Dr. Pei-Jer Chen as one of the leading scientists in the viral hepatitis research. So we know Dr. Chen have been involved in hepatitis B, hepatitis C, hepatitis D research, and actually, I just learned from Dr. John Taylor that previously I always thinking that Pei-Jer have to get his postdoc training with John, and actually, he gets his PhD on the supervision of Dr. John. And actually, his work on HDV identifies HDV RNA species in the infected cells and discovers both genome and antigenome circle and developed an assay to study HDV replication. And he left John lab. He continued work a while for the HDV and found that large, small delta antigen is required for RNA replication, and large delta antigen is for virion particle production, and also did small delta antigen phosphorylation and acetylation and their roles in viral replication. And in the HBV research, Dr. Chen have been, did a lot of very nice clinical studies and identifies viral and host factor that's related with HCC developments. And in the HBV virology, I think that one of his major work is that characterize this hydrodynamical HBV model that's refine what kind of construct you used and what's one strand you used, you can actually make it persistent replication or just present replication, and use that to figure out the immunological basis. Another very interesting work he did is actually figure out that some of the HBV's plasmid RNA species and their function in HBV pathobiology. And recently, we actually have something connected. A few years ago, my lab discovered that host cellular PP1 that is dephosphorylated core protein and required for the pgRNA package. Pei-Jer's lab actually discovered that how that PP1 is actually packaged in capsids. It's through interaction with polymers. That's just published 2 months ago. And in recent years, Dr. Chen have did a lot of very nice work on the HCC genetics and also developed an assay for the precision diagnosis and treatment of HCC. So without further ado, let's invite Dr. Chen present the seminar.
Thank you, Ru Tao, for the very nice introduction. Actually, when I I received the notice to deliver a talk in the Hepatitis B Foundation, I was a little bit nervous. So, but I'm become so relaxed when coming to the site, because there are so many old friends and also there's young newcomer. So I'm very happy and delighted to give my prospect and my observation about the hepatitis B. And it is the first half. The second half, I will update you some of the study going on my lab. These are very interesting questions, and I want to share with you. And before the presentation, I really want to thank Professor John Taylor. John is my advisor for the PhD study 40 years ago. So it's so unusual. We can 40 years later we can meet each other, and I'm very happy. John is healthy and active, and even coming to listen my seminar. Thank you. The talk of my today's talk, the bit is the elimination of hepatitis B. And actually, I want to take the experience in Taiwan, showing you how we can convert an epidemic become a rare disease. Despite all this treatment, this progress, we still have a very interesting challenge ahead. Not only scientific and also the medical. I want to share with you some of the point of view. Here is my disclosure. And again, we are deeply indebted to Professor Blumberg because his discovery of hepatitis B then laid down all the basis so we can study this intriguing virus, and also we can control the infection, elevate the health of the people worldwide. And here is the book for his autobiography. I recommend all the young newcomer to read this very interesting and fascinating history of his life. And this photo was taken about 25 years ago when Professor Blumberg visited Taiwan and giving a lecture at our hospital for the 100-year anniversary. And to start with the hepatitis virus, the story coming from the transfusion hepatitis. As you know, after the transfusion could be conducted with the knowledge of the ABO blood typing, then the transfusion has become a very important medical treatment worldwide. But however, before the year 1960 or 1970, after the transfusion, about 30% of the patient will suffer from the jaundice or hepatitis. So because this is transmitted through the transmission, so it's definitely an infectious disease. And at this time, they also filter the blood through this very small filter, and excluded the bacteria. So from this very simple study, the scientists know that the infectious agent were not bacteria, but the smaller infectious agent, likely the virus. So therefore, the search for the transfusion transmitted hepatitis virus or infection agent is a very important target, and also unfinished task. So many virologists and hepatologists working on this topic, but they all fail. But incidentally, Professor Blumberg, as you know, he's not a virologist. He's also not a hepatologist. But he's an anthropologist interested in the polymorphism, all the different immune response, from the different race, different tribe, how they after the infection. So his interest is to collect the serum sample from the different race or tribe of worldwide, then study their interaction. Through this, Professor Blumberg identified one interesting antigen from the native Australia native. So this is a very simple double diffusion assay. I guess most of the students they did not know this simple assay, but it's very easy and very reproducible. And if there's an antigen and here is a specific antibody, then they will interact to become a very specific precipitating line. And this antigen was Professor Blumberg named Australia antigen. And later, he worked with Dr. Thomas Landon and proved this antigen is responsible for the most of transfusion transmitted hepatitis. And here, actually, the Australian antigen later shown to be the HBS antigen subviral particle, as shown in this electron microscopy. So this is a very good example of the scientific serendipity through a different point of view, then coming to a discovery and resolving a very important medical issue. So the scientific curiosity and the discovery is still the ultimate goal for most of all the us. And through this, Professor Blumberg and Professor Thomas Landon also showed if we screening the blood donor for the HBS antigen, then excluding those passive donor, the transfusion transmitted hepatitis will reduce, the incidence will reduce from 30% to only about 5%. So this virus is really a major culprit for the transfusion transmitted hepatitis. And so this discovery impact the Taiwan a lot. This is far away from the Philadelphia, because for the long time, from the year 1902, when Taiwan is still in the Japan colonial state, at this era, and one university, a study from the one university hospital, they did the autopsy for the patient died in that hospital to understand the reason of the cause of the death and what is the reason. And they found in the Taiwan's population, the Taiwan patient died in this hospital, liver cancer accounting about 1.2%. This prevalence is much higher than the counterpart in the United States or the Germany, because in these two countries, the liver cancer only accounted for 0.1 or 0.2%. So in the Taiwan, the importance or the prevalence of the liver cancer is actually 10 times or 20 times more than USA and Germany. But there's a reason, we don't know the reason. At that time, there are many speculation. But however, after the discovery of the hepatitis B surface antigen, the Professor Song, my mentor's mentor, he studied the Australian antigen in the liver cancer patient, showing that most of the patients carrying in Taiwan carrying the Australian antigen. Later, he did an entire population survey from the young kid to the old people, and showing in the Taiwan the population at the age of 30, almost 90% of the population were infected by the hepatitis B virus, and among them, about 10 or 20%, they become the chronic carrier. So the prevalence, this prevalence is very high. If you think about the diabetes is only about 5 to 7%. So, but the hepatitis B is much higher than diabetes in Taiwan at that time. So this is really a very serious problem. So the Professor Song decided to study this and also convince and trying to identify how to prevent it. And the reason for hepatitis B continue to perpetuate in the Taiwan society is because of the perinatal infection. For the mother in the Taiwan, the childbearing mother, the HPV prevalence is around 50%. But during their delivery, in the perinatal infection, that most of the children will to be infected and become chronic carrier. So when these children growing up, they become the mother and they carry out the entire life, the cycle of the infection in the society of the Taiwan. Not only the Taiwan, actually in many parts of the China. In order to disrupt this cycle, the most important way is trying to block this infection by vaccination at the very immediate after the delivery. But unfortunately, Professor Blumberg also is so insightful. He originally proposed the surface antigen subviral particle can be adopted as a vaccine, because this particle, they don't contain the virus DNA. So if we purify this particle and after appropriate inactivation, it will become a very useful vaccine. And then his hypothesis was confirmed. And so our first generation vaccine, hepatitis B vaccine, actually coming from the plasma. And then it's so far, the vaccine is very effective, was effective and very safe. But the data they are replaced by the the vaccine produced from the East by the recombinant DNA technology. And because the Taiwan, the hepatitis is so prevalent in Taiwan, so Professor Song, and he is another my mentor, Professor D.S. Chen. Professor D.S. Chen was also the Blumberg Prize winner a few years ago. So Professor D.S. Chen, he masterminded that universal HPV vaccination in Taiwan, and after a very long, a few years preparation work, finally we start the HPV vaccination to the new around the year 1984. This vaccination continued until now. But the efficacy is very impressive. In our 2014 survey for the population about the HBV S antigen carrier prevalence. For the population younger than 30 years old, this population were the vaccinated cohort, and the HBV S antigen carrier rate average is only 0.5%. But for the older than 30 years, these are the pre-vaccination cohort. The HBV prevalence is around 7%. So the difference is is so much. So from the it's a 90% reduction of the HBV antigen carrier rate after the vaccination. This is very important. And not only reducing the HBV antigen carrier rate, but also the vaccination again further reduce the childhood liver cancer because after the vaccination, the children almost free of HBV infection, and their childhood liver cancer, the rate dropped very quickly. So this is a very illuminating case showing if we prevent a carcinogenic virus, we can the infection of carcinogenic virus, we can prevent the cancer. The cancer related to the infection. And finally, the HBV vaccination data spread to almost every country in the world, and you can appreciate. See this red area are high prevalence area, but after the vaccination, the post-vaccination cohort, like China and the East Asia, the HBV antigen carrier rate rapidly declined. Now the most difficult part is now is the African area. They are still very challenging to implement and continue the HBV vaccination program. So the HBV vaccination is really the main pillar in controlling HBV infection worldwide. So it is important to maintain and consolidating the HBV vaccination. For example, in this here is the case. In France. In France, the HBV vaccination uptake rate is only about 60%. But at the one time, there is one misinformation linking HBV vaccination to the multiple sclerosis. So with this misinformation, the vaccination uptake rate in the French dropped quickly. But at the same time, usually this is acute hepatitis B in France. After the vaccination, it declined a little bit. But when the young generation dropped the HBV vaccination, the acute HBV infection quickly rebound. This is just showing you how important the HBV vaccination is to be maintained. And this is become very challenging. And in the era of the vaccine hesitancy, we still have a long way to go. So we already almost resolved the HBV infection in the vaccination cohort. But however, for the pre-vaccination cohort, now there are still about 250 million hepatitis B carrier worldwide. This is a report from the WHO. And for this patient, if they don't receive any active antiviral treatment, they may progress from the chronic hepatitis to the liver cirrhosis and to the HCC. And the lifetime risk for the carrier to progress into the end stage of liver disease is about 10 to 30%, depending on the gender. So this is very significant. If you think about, we have 250 million, and if there's only 10% of the patient with died from the end-stage liver disease, we have 20 million. So this is also a very heavy disease loading. Therefore, the treatment for this population is really urgent. And the treatment become possible because of the two scientists, Professor J.C. Summers and the deceased Bill Mason. They discovered hepatitis B virus, though the virus containing HBV DNA inside of the virion, but actually, after the infection, the viral DNA will repair, converted into the cccDNA, and then produce the pregenomic RNA. The pregenomic RNA packaged into the capsid. And before the mature virion, the RNA will convert into the cDNA. Then later, released into the mature virion. So HBV is similar to the retrovirus. Its life cycle involves the reverse transcription. I still remember when I visited John 40 years ago. He gave me the paper to read. It's very difficult, very difficult paper to read. I spent about 2 weeks to read this article carefully, but I learned a lot. Because of this, because the HBV replication through the reverse transcription, therefore similar to the HIV, but there are many drugs already targeting to inhibit the reverse transcriptase of the HIV. Therefore, this drug can be quickly applied to the hepatitis B. So the scientific discovery really shortened the timing for the effective antiviral for the hepatitis B. And the reverse transcriptase inhibitor broke at this stage pregenomic RNA to the DNA. So the continuous intake of all the drug will stop the production of mature virus into the circulation. So if the chronic hepatitis B patient taking antiviral therapy for sufficient time, then the HBV DNA in their blood become undetectable. And important, they are almost eliminating hepatitis B activity. So finally, even in some cases, they can reverse the liver fibrosis. And there are many clinical long-term studies showing the long-term taking of the nuclear antiviral therapy can reducing the progression into the end-stage liver disease. They are about 50%, and also reducing liver-related deaths, the 70% reduction. So this antiviral is very safe and very effective and very simple, just one pill, one day. And I also want to show you the situation in Taiwan. So after the HBV vaccination and the HBV and HCV antiviral therapy adopted in Taiwan, since the year 2003, the liver cancer incidence, here is the incidence of the male dropping, decline steadily. This is the same thing for the female. So the treatment, the vaccination and the antiviral treatment is become effective, and the liver cancer incidence in the Taiwan starting to decline. Now it's almost every year decline about 3 to 5%. And then at the same time, the treatment not only reduce the HCC incidence, but it also reduce the deaths due to the chronic liver disease or the cirrhosis. This is a so-called hepatic failure. They're not related to the HCC. I showing you the situation in Taiwan. In the year 2003, the end-stage liver disease ranked number 6 of the 10 leading causes of the deaths in Taiwan. But the data it dropped to 7, 8, 9, and 10. Now the year 2020, the end-stage of the liver disease ranked 12th in Taiwan. If you compare the situation in the United States, here is the chronic liver disease, end-stage of the liver disease. In the year 2019, it's about 11, but now it's increased. It's jumped to number 9. So we are doing in the Taiwan, the end stage of the liver disease, the priority is really going down, but in the United States, it's going up. But despite all this achievement, we still face a lot of challenge, especially for the hepatitis B. We follow here is the HCC incidence divided by the HCV or by the HBV. I want to show you this is I draw your attention to the slope. Here is HCC incidence. You see there's this slope for the HCV related HCC, it dropped pretty quickly. But for the HBV, it's almost flat, very limited. So the major there are a few reasons, but one of the major reasons is the hepatitis B patient, they are only small proportion receive antiviral therapy. Here is a worldwide WHO treatment cascade. Actually, only about 10% of the hepatitis B patients, they are eligible for the antiviral therapy, actually receive the treatment. Even in Taiwan, we have only about 30% of the patients receive the treatment. So what the major reason is because the treatment guidelines are too complicated. The treatment guideline usually proposed by the Hepatologist Association. So it's very complicated. They need to check HBe antigen and also need to check HBV DNA, ALT, fibrosis. This complicated criteria can only implemented by the hepatologist, and it's very intimidating to the patient and also to the internist. This is a major reason deterring the doctor in prescribing antiviral therapy. So WHO fully aware of this the barrier. Therefore, they are in their most updated version of the guideline for the hepatitis B treatment. It's put the criteria very simple. If the patient has significant fibrosis or they have other comorbidity, then they can be treated. You don't need to check the E antigen, viral DNA, etc. So it's become very simple and very accessible criteria for the treatment. I think this is very important. Otherwise, we have so many hepatitis B patients left out in cold. They cannot receive treatment. And one very interesting and important case is actually to consider the universal treatment and on-demand treatment. Here is a very interesting study from Hong Kong. They compare, the liver cancer incidence, accumulated incidence in the HIV and HBV co-infected patient or hepatitis B alone patient. Because for the HIV and HBV co-infected patient, they immediately receive antiretroviral therapy. So the retroviral therapy also control the HBV. So for HIV patient co-infected with HBV, their HBV is treated universally. They don't care about any their HBV status. So this is the universal treatment. But for the hepatitis B alone, they are actually they only receive treatment after they reaching the treatment criteria. And then the long-term follow-up showing the HCC incidence. Here is the HBV alone. This is HBV co-infected with HIV. This is clearly there's the HCC incidence in the HIV and HBV co-infected group is much lower. So this is another piece of evidence calling for universal treatment. But we also are working on the Taiwan's data to see whether we can reproduce their result. But if this is true, this would be a very interesting point to consider to treat the hepatitis B patient in the future. And the treatment and the antiviral treatment, another very important change is the antiviral treatment will convert the very complicated natural history of hepatitis B into a very simple one. So you see that there's a DNA hepatitis and they are fluctuating in the life of the chronic hepatitis B patient. But this can be very simplified after the antiviral treatment. If we continue the antiviral treatment, the viral DNA drop become undetectable. There are no hepatitis. And the virus surface antigen, E antigen, declines slowly. So it's very simple. What the patient and the doctor need to pay attention is to ensure the adherence of the NA therapy and the drug, note the drug resistance. And then the second, you can stop the NA therapy when the HBs antigen is cleared. And then the third, we survey for the cirrhosis and HCC. So the treatment management of the chronic hepatitis become very simple. You don't need to know E antigen, what the antigen or HBV DNA level. You just forget it. Just treat it. Treat it. Although the treatment, NA treatment cannot cure HBV in most of the patients. But they are very useful and they are accessible to the majority of hepatitis B patients. In order to cure, now so far, the best approach, the most promising approach is gene editing. And we're also working on this. But however, the gene editing is scientifically fantastic. But I don't think even it's successful. It can, because it's very difficult to implement worldwide. It's probably only in the advanced, in the more developed country. For the majority of the hepatitis B patient, this is only probably is is a far away. The most practical is still the NA therapy. NA therapy, they probably are more important to implement. So I give you my review about the vaccination and the antiviral therapy for the hepatitis B. We are making progress, but we still have some long way to go. So I want to share with you some of the scientific study or clinical study working in my lab. So the first, the HBV splice RNA. As you know, the HBV transcription conventionally, we know there are 4 contiguous messenger RNA. This one is for the core and the pole, the pregenomic RNA, and the 2 for the large S, middle S, and for the X. But however, in the 1990s, we and others discovered the pregenomic RNA can undergoing splice and generate a series of the splice RNA. But we don't know what is their function. and then the most abundant one is this one. This one is called splice SP1 splice RNA, accounting for 50% of the splice HBV RNA. So the biology of the splice RNA is kept in my mind for many years. But finally, we have experiment data showing that the major splice RNA SP1 actually encoding a novel core protein important for the infection. Here is a traditional HBV particle. We know that the virus core protein, and together assembly with the proteomic polymerase and the surface protein become. So this is the classic, the conventional HBV component, the virus structure. But however, we have shown this virus particle actually poorly infectious in the human hepatocyte chimera mice. They needed another protein encoded by the SP1 splice RNA. This SP1 encode a protein is almost identical to the core protein. This one a derivative tension. The original core protein is 182, and ended C terminal is 16. But the SP1, because of the splicing, it just remove the single cysteine. And then this protein we have shown it's also incorporated into the virus, and only the virus, the core protein incorporated not only the core 122 but also 121. Only this virus become quite infectious, very infectious. But this one is poorly infectious. So it's interesting, but I think it is worth to further study the HBV splice RNA. Maybe we can find the new antivirus or new immune target in the future. And the second topic I'm still working on is the HBV, the persistence of the HBV infection, because the HBV, the HBV clearance is very important. It depends on the age of the infection. Here is a very famous study published by the McMahon and also the Dr. Palmer Beasley. They already show if the children infected at the birth, especially the perinatal period, the majority of the kids, they cannot clear the virus. 90% or 95% become persistent. But in contrast, if they infected after the 5 years old, the majority of the kid or the toddler, they will clear the virus. Only 1 or 2% will become chronic. So this is very interesting, because if you look at the hepatitis A infection, all the subjects clear the virus, no matter what is the age of the infection, irrespective of age of the infection. For the hepatitis C, it's just vice versa. Majority become chronic, but only hepatitis B, the clearance rate depends on the age of the infection. So what is it? And we are very interested in studying this phenomenon. So I spent we established a mouse model. You know that mouse cannot be infected by HBV. So we generated a plasmid. This plasmid, we can inject this plasmid through the tail vein into the mice. And then this tail vein injection, we deliver the HBV DNA to the liver as the most important target site. So after the DNA entering into the hepatocyte, they will produce viral protein, HBV protein, and the virus, and inducing immunity. And Professor Tresari showing in this situation, in this experiment design, if you injected the plasmid into the biopsy, the mice animal clear the HBV in surface antigen within 2 or 3 weeks. But we found that if we inject this plasmid into the different mice, especially the C3H or the CBA mice, if we inject the mice at a young age, for example, 6 weeks old, the majority of the mice will become persistent for the hepatitis B. But if you inject them at the age of the 12 weeks, the majority of the mice will clear the virus. So we can duplicate, reproduce the age-dependent clearance in this animal model. And further, we are showing this is related to the development of establishment of the gut microbiota. Because these mice, they are syngenetic. So their background, genetic background the same. So the difference is only must be environment. And then we show that the gut microbiota is very important. And recently we we further define showing for the C3H mice, if we expose these mice with HBV as a germ-free mice, they remain HBV persistent. But in the SPF mice, some of them will clear the virus. But recently, we identified a single species of the bacteria. This bacteria, if you give the mice earlier, they will clear very, very quickly. So now we are working on this, how this specific strain of the bacteria, specific strain of the bacteria can help clear the HBV in this mouse model. And the next, the third, I want to talk about my favorite virus, the hepatitis delta virus. This is a very small virus, but it's so interesting. I just cannot stop to telling you the story. So this is a very small, only 1.6 kb circular RNA. When I started with John 40 years ago, but we're still working on this. It's only containing one operating frame encode the virus delta antigen. But if you put this delta RNA into the cell, they can replicate, but they cannot go outside the cell. If they need the hepatitis B surface antigen to package the delta genome and the delta protein to become an infectious virus. So delta virus depend on HBV for the transmission, but not for the replication. The circular RNA has to go through a very different cycle. We have been very interesting how they replicate, because the HBV encode protein is delta antigen, but this delta antigen is not RNA polymerase. So they must borrow the cellular RNA polymerase for the replication. But so far, we have not succeeded in identifying or showing that definitely which RNA polymerase is responsible for this approach. I'm still working on this, not yet making progress. But however, we did show that the delta antigen, because this different step, they all need the delta antigen. But how delta antigen recognize the different stage of the HDV life cycle, and we have shown the post-modi the phosphorylation or the acetylation probably can render the delta antigen to carry out their function at a different stage of this life cycle. Organs. So now we're running out of the battery. So I try this one. So this is the phosphorylation acetylation probably working in this area, in the separating the different replication stage of the HDV. In addition, one of the PTM, the isoprionation of the large delta antigen, responsible for the HBV packaging. So this is also become a target for the antivirus. Dr. John Glenn, he developed the lonabenib to inhibit this PTM and then stop the HBV packaging. And recently we are interested about the RNA modification, because the delta RNA is also can be modified, because the many cellular RNA modified by there are many RNA modification. The most abundant is the M6A modification. It modified the methylation at the adenine number 6. And this carry out the cellular METTL3 and MATTL14. So we are asking whether the M6A modification playing role in the delta life cycle. So we generate one METTL3. This is the enzymatic molecule for the M6A modification. We generate this knockout cell line, and we translation HBV DNA, HBV cDNA, and HBS antigen to see their packaging. In the wild type, their norovirus can be packaged. But if you knock out METTL3, the packaging drop to only about 5 to 10% for the wild type. So therefore, the M6A somehow modulate, especially the HDV packaging. They don't have effect on the HDV replication, only in the packaging. So this paper is still in revision. So I just tell you our interest in the hepatitis B and the hepatitis delta. And finally, I want to show you a very important topic about the liver cancer. For the advanced of the HCC, for a long time we know the chemotherapy is not effective. This has been known for many years. And in the last 10 years, there are some molecular targeted therapy, such as the sorafenib, the imatinib, for these are the multiple kinase inhibitor. It's been shown effective in control liver cancer. And recently, the anti-PD-1 and anti-VEGF also showing more better efficacy. However, I want to draw your attention for this approved drug for the advanced HCC, actually not developed specific for the HCC. They all developed initially for the other cancer. For example, sorafenib is developed for treatment of the renal cancer. But because it's effective in the renal cancer, so they try to in the HCC. Fortunately, they did see some effect. And similarly, this one is initially developed for the thyroid cancer. And this combination for the lung cancer or colon cancer, there's nothing specific developed specific for the liver cancer. So the response rate is usually limited. Only 20%. Only the best one is close to the 30%. So we are interested to see whether we can find the driver mutation for the liver cancer. And in the case in the HBV related HCC, the most common somatic mutation is HBV integration. It happening in about 80 to 90% of the HBV related HCC. Here is the classic southern blot, and the integration is also very interesting. The integration to the liver cancer chromosome by the HBV DNA happening in the random. So for individual HCC, the integration site is unique. There's no two HCC, they can share the same site, exactly the same site. So it's different. And And recently, we studied the HBV integration into the HCC, and discovered we can divide. We identify 3 independent mutations. These mutations, they are mutually exclusive. One mutation is the HBV integration into the third promoter. This taking place in about 25% of the patient. And then the other is the HBV integration into the exon 306 or the KMT2B. This is the histone lysine transferase. This is about 15%. And the third mutation is the third promoter mutation. This is minus 123 mutation. All these 3 mutations, they can be identified in the HBV-related HCC, but they are mutually exclusive. So we can divide HBV-related HCC into 4 groups by the 3 mutations, 3 somatic mutations, and the other group are free of the 3 mutations. And this mutation, they have different driving force. Now we are trying to see whether we can duplicate this HCC model in the mouse. So we are trying to overexpress the third. This is the telomerase and all the KMT2B in this mouse liver and generate the corresponding HCC model in the mice. Therefore, in the future, we can develop specific anti-cancer therapy. And another point I want to share with you is the HBV integration. I just mentioned to you for individual HCC, their HBV integration is unique. Therefore, the junction cover spanning HBV sequence to their junction covering the HBV sequence and the human sequence. This sequence is unique. We call it viral host chimera DNA unique to individual HCC. This fragment, if they can be released into the circulation, so they become a useful biomarker to tracking the HCC. So now we are applying this the detection of the chimera HBV DNA in the HCC to follow their treatment response to the anti-cancer or their minimal residual disease. But in the future, because of this clone, they are actually started from a small clone and then gradually evolve into the cancer. But their chimera DNA, if they are the same clone, they remain the same. So we can detect very early. So in the future, I think this is chimera DNA will be very useful in tracking the clonal expansion or the liver cell into the HCC. Though our sensitivity at this stage is not so good, but we are working on this aspect. Therefore, in the chronic hepatitis B, when progressing into the cirrhosis or into the HCC, usually the HBV DNA level is declining, surface antigen is declining, but however, the chimera DNA is probably rising. So we can combine alpha-fetal protein and the chimera DNA to track or monitor the development of HCC. Finally, I want to share with you a very interesting clinical study. For the early stage of the HCC, after the surgery, the problem is HCC recurrence. In the first year, there are about 25% of the recurrence. So the adjuvant therapy is very important. And here, this is a very famous STORM trial. The PI give the sorafenib after the HCC curative surgery. And see the drug can prevent recurrence. But unfortunately, the treatment and the placebo they are the same. The recurrence rate, they are the same. So the drug, the trial failed. But recently, there's an anti-PD-1 and an anti-VEGF. They treated the patient after the surgery for 12 months. And then in the 12 months, you see some significant difference. But if you follow data, you don't see any difference. So this is still a challenging issue for the post-curative therapy. And a few years ago, we started a new therapy by using long-acting interferon followed by the anti-PD-1. I just put a long story short. We treated the HCC patient after the surgery with this formula of the adjuvant therapy. Very, very surprising. So far, we treated 15 patients, but none of the recurrence so far. So this is we are trying to expand into the further study, but this is a very encouraging and a very interesting development. Finally, I want to share with you, this is a hepatitis B, because they have already locked in human history before the vaccination. It's for many years. See, this is the HBV identified from the Korean mummy in dated back to 19th century. And then, you know, last 2 years ago, they also found the HBV DNA in the hair lock of the beetle. So the greater composer. So the chronic hepatitis B already in the human for many years. But after year 1984, after the vaccination, the carrier rate dropped quickly to the 0.5%. I think in the future, another 20 years later, its profit dropped to 0.01%. So this is very close to a rare disease. So I think the Professor Blumberg discovery already converting an endemic to a rare disease. Is finding preventing millions of infection and saving millions of lives. I think he is really greater future in the scientific or medical history. But however, can we eradicate hepatitis B? We know that the only successful case is a smallpox. Here is an emperor from the Ming Dynasty from the China. You see there the pox cup over his face. And Professor Henderson did worldwide effort eradicating smallpox. But however, when in his obituary, he suspect this is probably only the one successful case. For example, even the much older time for the polio, they have been 7 decades, but we cannot eradicate polio. So that in the evolving and the changing social infrastructure, and also the hesitancy in giving vaccine, I think the eradication would be very challenging. Finally, I want to show you the legacy. Here is Joe and me taking a picture in Seoul in year 2016. And in this picture, Henjong visited Taiwan in the 2017. So, and there are many students who are coming from Jones Lab. And here is the Fox Chase Cancer Center. I think the Fox Chase Cancer Center was a mecca for the hepatitis virologists 20 or 30 years ago. And I hopefully maybe Hepatitis B Foundation or Blumberg Institute become the mecca for the future virologists. So I began my PhD student study with John in the year 1983. So it's almost 42 years ago. So afterward, I moved back to Taiwan. And unfortunately, guided by Professor Song, Professor Pei-Jer Chen, and working on this area. And also I generated my next generation. I generate, and we are still interested, not only in the medical or the prevention of hepatitis B. The virus is still very intriguing scientifically. It's so interesting. Hepatitis B, hepatitis data. So I encourage the young generation to study the two viruses. There are still many unknown we needed to learn. I just want to share with you the enthusiasm for these two viruses. And also I want to thank the Hepatitis B Foundation for the Blumberg Prize. Thank you so much. Thank you. Yes, Steve.
Yes, we'll stick around a little bit for some questions. And
Yes.
people may have to go. I know there's a reception. First of all, thank you very much. Great talk, really enjoyed it. And I wanted to say before I ask any questions,
Yeah.
that John Taylor is in the audience. And also is Chris Seeger, two other Blumberg Prize recipients. I don't know if anyone else is here who won the Blumberg Prize. But John Taylor was Christoph.
Yes, Chris.
Oh, Christoph.
Yes.
Blinking at his feet. All right, so thank you. Okay, a very general question. You mentioned vaccine hesitancy a number of times
Yeah.
in the end, and I appreciate that. Are you seeing vaccine hesitancy, one in general in Taiwan now, more than before, and to the hepatitis B vaccine?
For the hepatitis B vaccine, I don't see the vaccine hesitancy in Taiwan.
Oh,
Because it has been implemented for almost 40 years without, and we also have carefully monitoring any adverse effect. So this is okay. But for the other vaccine, for example, the human papilloma vaccine, we see the protein vaccine hesitancy in Taiwan. So this is, I think. So this sooner or later, I'm worried whether it's become a problem.
Other questions?
Professor Chen, despite the fact that Taiwan is excluded from the WHO, I know you're committed to meeting the eradicate hepatitis goals by 2030. I have seen that you're doing pretty well against Hep C. How are you doing against hepatitis B?
You mean the hepatitis C or the vaccine?
Hepatitis B, how are you doing against the WHO goal of eradicating Hep B as a public health threat?
Oh, I have to say we have a long way to go.
Yes.
I don't think we can meet the SDG for the hepatitis B by the WHO. There's especially for the treatment. For the vaccination, we are doing fine. We meet most of the goal. But however, for the treatment, prevention or the death or reducing the HCCs and the death, we cannot make it.
So they may, the biggest challenge might be identifying Hep B infected individuals.
This is not the major issue. The major issue is the treatment. It's a barrier. It's a barrier. Because in the Taiwan, we have the hepatologist controlling the guideline.
That single goes back to your slide that you showed.
Yes. This is a barrier. We are fighting for this barrier. And unless we, then we can treat more patients, more patients. I can tell you, for example, in the Taiwan, we have only 200 hepatologists, but we have 1.5 million hepatitis B carrier. It's similar to the United States, but the United States they have about 10,000 hepatologists. So in Taiwan, if you rely on the hepatologist to treat the patient, it is impossible. even 10 or 20 years. It's still long way to go.
Are the treatments covered by insurance, health insurance in Taiwan?
Insurance is not a problem.
That is not.
Not a problem because the drug price can go down very quickly. We can easily expand the coverage 5 times, or even 10 times. But the problem is you need to go through the medical system.
Yes.
So this is a problem. This is a problem. We have so few pathologists, so few specialists. We cannot manage so many patients.
Thank you.
Yes.
Me,
Yes, you can.
PJ, always enjoy your talk, wonderful. And follow the comment, I think you propose a Hep B treatment, like an antiviral treatment.
Yes.
100% behind that.
Yeah.
And like you simplify logic, then it's just antiviral treatment, universal.
Yeah.
And you can have either ID people or the general physician. I think that's wonderful.
Yeah.
And hope you can fight that for it. And now I have I also very intriguing about your research on mouse model
Model, yes.
about the agile infection, like parallel to the human,
Yes.
according to your observation. And then you find out that the certain bacteria strain play a role. I wonder, have you thought about the gut microbiome? Compare those different age, the human do have those microbiome data.
Yes.
And so you mouse model the pneumonia and then those group, and you compare their microbiomes, bacteria population. And then maybe we can discover something.
Yes.
And I think that's very interesting. Besides, you discover a mouse model can be infected by HPV. That's fantastic.
Not the infected. I have to say, this is a transfected system. So it's a HPV persistent, not the infection.
Persistent. Yes.
And we did find one bacteria making significant difference in the mouse model. But now we are trying to see whether this bacteria also working in the human, but not yet there. Now, where I cannot tell you.
You might find something compatible.
Yes, we think, but
That's interesting.
yes, we are trying to see whether this specific strain of the bacteria making difference in the human. But I cannot tell you. I'm not sure. Because the human is much more complicated. Much more. I cannot.
The certain gut microbiome, many study was pointing to the human immune response
Yes,
or
yes.
immune development. So
Yes, this is the idea.
Yeah.
This is the initial idea. So for the mice model, we are just trying to see whether we can apply this specific bacterial strain to understand how HBV is cleared. But I cannot say our finding in the mice model can be directly applied to the human. Okay, because this is a different situation. Okay. Yes, and you know.
Yeah, Dr. Chen, I'd like to ask a question about HDV therapy. Treating HDV infection seems to be considered as a lower hanging fruit for the HBV entry inhibitors because HBV seems to have higher dependency on infection of new cells compared to HBV. What do you think about the feasibility of developing antiviral targeting the intracellular life cycle of HBV, the genome replication?
I do because if you think about the virology, all the DNA virus after the infection become chronic, unless only some of the cytolytic virus like the adeno, they don't become chronic. Otherwise, almost everyone become chronic. So DNA virus is very difficult. But the RNA virus, except the retrovirus, all the HCV, all the all the delta, they don't become chronic. Okay, so I think the HCV is also a very good example. If you broke the viral replication, the cellular innate immunity can clear all the intracellular viral RNA. So for the HDV, I think it's maybe it can have, but so far, but now we don't have the antiviral can stop the HDV replication. Our drug is only inhibit their packing or inhibit their entry. We don't stop their replication. Even the RNA, I don't see very very promising direction. The fundamental, we need to know the polymerase. We need to know which polymerase and how they interact with the delta and to design a small molecule to stop the replication.
So let me, because the time limit. So for the Blumberg faculty and research staff, we're going to have a roundtable discussion with Dr. Chen tomorrow afternoon.
Well,
So just leave the question for other people.
your question.
The delta replication can last 6 weeks without any help from hepatitis B.
Yes.
And the N6-methyl that you're talking about, I mean, what other effects are on a cell if you suppress N6-methylation? I mean, can you have a positive control to make sure that you're not doing a lot of damage to the cell rather than just the delta?
Yes, you're right. I cannot say so for the inhibit inhibited the post-translation so far is not working. Okay, it's not working. Oh, it's more complicated. It's more complicated. So I don't think that if we targeting the antiviral for the protein post-translation modification, I don't think it's viable. For the inhibited RNA modification, I also don't think it's viable. Fundamentally, we still need to know the replication. Okay, you're right. Yes. Yes.
I ask another silly question.
Yes.
A lot of viruses that have DNA genomes that undergo splicing accumulate small RNA circles.
Yes.
If you're so convinced that splicing occurs for hepatitis B, then there should be small circles that can accumulate
Yes.
that contain only viral sequences.
Yes.
Why haven't they been found?
This is also a very good question. But some of the intron can stay in as because after the splicing you will see the intron.
There's lots of examples, but is there any examples for hepatitis B? There, you have evidence of the introns surviving and accumulating?
No, I don't see the intron survive.
No.
The majority that we don't see the intron survive. Okay, but recently we did find some very intriguing small, very small. It's just beginning. I was very surprised. We did find some small HBV RNA, quite small.
But not circle.
I don't know whether it's circle or not. I'm still working on this. This is just coming out only a few months ago. Yes.
Yeah, I was curious, does this one about the slice variant, does that get incorporated into HDV, the HDV virions?
Yes.
It does. The SP1, does that get incorporated
Into
into
the delta, you mean the
HDV? Yeah, into the delta.
So far, I don't know.
Okay.
I don't know. I did not. Yes, I probably because the SP1 is a core protein. It's a core protein. So for the delta, I don't think it's required. It's only needed the HBV surface protein, not the virus core.
Got it. And have you looked for perhaps some of these splice variants affecting co-infected viruses? As a function, if you haven't been able to find functions for them? Yeah.
So far, we just find the SP1, the function of the SP1. But when the spread, when it influences the HPV viral infection, I'm not sure. We are looking for another spread. And the data is really controversial, so I cannot give you my David idea. So, all right.
One last really
Yes.
sort of higher level question.
Yes.
So I'm a drug developer.
Yeah.
You're recommending that, and you showed really great data saying, if you treat with antivirals earlier,
Yeah.
right, you can really lower the incidence of liver cancer. So if that became the way we treated patients, how could we make better antivirals for HPV? Yeah. What, if we look beyond getting the physicians to change.
If you take the case of the HIV, we need the HIV very similar to the HPV. Initially, they needed to take the pill every day, right?
Right, entecavir or tenofovir.
But now they developed the very long-term activity. The Gilead now developed a very long-term small molecule working for 6 months. I think if in the future, if we can develop the antivirus, you give the antivirus every 6 months, this will be much better in terms of the heals, the antivirus, and also the prevent the infection. So I will encourage you to think about this. In fact, for the HIV, the new drug is disrupting the core assembly, like the CPAM in the HPV. But the HPV CPAM is not so long acting. The HIV is very long acting. I think in the future we needed the long acting reverse transcriptase inhibitor or the viral replication inhibitor.
Thank you, Dr. Randy. It's a really excellent presentation. I want to dive in a little bit more about the eradication you talked about. I mean, Taiwan is clearly the front lines of the HPV and has been for decades. You've done amazing work. We have a highly active vaccine that has such a pronounced effect on reducing incidence and incidence of cancer. And we have your data about treating patients who have the virus to prevent both disease and infection. So why not just do it? And vaccination compliance in Taiwan at birth is probably very, very high, I would assume. And the vaccine is highly effective.
Right.
So why not just continue that, screen and treat all these people, all the adults who have the virus, vaccinate those who don't. What in terms of elimination of the disease, which has been called for by WHO, has been called for in the United States by the National Academy.
Yeah.
Which one your thoughts are on that plan? I mean, does that sound?
You mean the vaccination?
Yeah, vaccination coupled with screening and treatment, because the treatment you said is effective.
Yes, this is the strategy that WHO advocate. The problem now is the treatment. Is the treatment, because the treatment is, the guideline is proposed by the hepatologist. Okay, so they are interested only in the patients, this patient's liver disease. They don't think about the infection, the transmission. For example, they objected, treated the young adolescent, young hepatitis B patient. This patient, because they don't develop hepatitis, but they carry rotavirus. But these young people, this young carrier, they are source of the infection. But the hepatologists, they don't care. They don't care because they treat this patient, they think it's not effective. This is totally wrong. Because if we are control disease, not treat a single patient. So this is different the point of view. All the hepatologists versus the infection disease. I think that's my point of view. So you are right. If you coming from the common sense,
Mhm.
this is okay. If you take a side of the case of the HIV, this is the right way to go. But what we face is the conservation, is the conservation of the pathologist. Okay? So I'm sorry.
I just have one follow-up to that. So the question would be, so why doesn't Taiwan move? So the United States is trying to move the treatment of chronic hepatitis B from hepatologists, because we don't have enough, to primary care. So one thing that is in Taiwan's favor, it sounds like insurance isn't a problem. the cost.
Another problem.
So why limit treatment from hepatologists? Why not let the primary care doctors?
This is why we are fighting for, because in the Taiwan, we have the pathologist objective.
They're objecting to
They
that.
are objecting that this is the treatment. This is a problem. You face the AS or DE, so the same thing.
Oh, but there's no money in
This money.
prescribing the drug. They
This is
do the annual physical.
This is not the money problem. This is the arrogance of the pathologist.
So it's territoriality.
Yes.
I don't understand because from your slide,
Yeah.
it appeared to be quite obvious that the young people in Taiwan
Yeah.
are vaccinated do not have HPV, right?
Right.
So now we're talking about people over 40. right?
Older than 40.
Yeah.
Yeah.
And they should all be candidates for
Yes,
treatment.
yes, this is the common sense, but the problem is the territory of the pathologist. You know, if you look at the ASLD, you look at the ESO, they are still a strong reservation. This is unfortunate.
To comment on the HPV vaccination, we have several people coming back.
They should have symptoms, right?
It should be HPV vaccination is part of travel EPI program. So every child will receive those vaccines in all those schedules, including at birth, the HPV shot. So because it's already accept universal EPI program for every children, so it's much easier to implement and everyone accept. And I still think treatment Taiwan is in the best position because money is not the issue. So PJ, you can do it, you have power to get a policy change.
I can give you the example for the hepatitis C. Now hepatitis C treatment is every doctor can prescribe. So we are pushing for the same thing for the hepatitis B treatment. But we face some barrier. But anyway,
You get the people to go to Taiwan and then support your policy.
We want to give a chance just to one person who hasn't, who's
Yes,
had
yes.
raised for a long time. And then-
Dr. Chen, thanks for the presentation. I was very curious, your adjuvant treatment therapy caught my attention, the nivolumab, and I think it's interferon that you're doing. So any speculation on what you think is a mechanism, the reason why, if I saw well?
it's very simple.
Yeah.
Because the current adjuvant, anti-VEGF or anti-PD-1.
Yeah,
This one is working on the adaptive immunity.
yes,
They don't touch innate immunity.
okay,
So our hypothesis is if you trigger ropaque interferon, it's the innate immunity followed by activating adaptive immunity. Maybe you do something different. So we tested this in the animal model and showing the sequence is effective. But if you put the reverse, it's not effective.
I see.
So that's our logic. Okay, but I'm not sure whether this is really so impressive if we expand the trial. Okay,
It's very impressive.
it's too impressive.
Yes.
I'm
Almost too good to be true.
It's too impressive. So this paper is just published, but I have to say I think it's too good to be true. And
Yeah.
No, I'm saying the reason I ask is we're a young company, we are growing and we are in the adjuvant area. So that's why I asked,
Right.
kind of.
You can use, I can send you the paper.
Excellent, thank you.
Because the logic is very simple. You activate not only adjuvant, because many not only the adaptive immunity, because now there are many anti-PD-1 and there are other combination therapies. They are all working on the adaptive, not the innate. So this is the idea. Okay.
Thank you.